What's wrong with me?
Are you alright?
Yes, just some trembling, that's all.
Give me some. Hey, they have this
What?
spinach. This iron can reduce your chance of Parkinson's, man.
Parkinson's? I don't have Parkinson's. I'm young, and besides, my PINK1 and Parkin are working quite fine.
Just eat it.
Do I eat it raw?
Mitochondria are organelles in the cell that is a site of cellular respiration. They are the cell's powerhouses. They release energy called adenosine triphosphate or ATP from the assimilation of nutrients and oxygen via a process called oxidative phosphorylation. This energy is in turn used to drive essential biochemical processes in the body. Mitochondria are found in every cell and are more abundant in specific tissues where significantly higher amounts of energy is required, like the muscle cells.
So, what happens when a mitochondrion becomes defective beyond repair? Embedded on the mitochondrial membrane are 5 complexes: complex 1, 2, 3, 4, and ADP synthase, which generates ATP from ADP and inorganic phosphate. At the same time, ROS are produced, and its accumulation will result in mitochondrial damage beyond repair, resulting in its eventual removal from the cells. This process is known as mitophagy.
How does mitophagy occur? Based on the PINK1 Parkin pathway. The pathway to mitophagy involves 2 proteins, PINK1 and Parkin. PINK1 identifies damaged mitochondria and targets it for destruction. This is done by recruiting Parkin, an E3 ubiquitin ligase which transfers ubiquitin to the defective mitochondrion. Subsequently, the proteasome recognizes the ubicinated mitochondrion and digests it, thereby removing it entirely from the cell. This process is important in ensuring that the defective mitochondrion does not interfere with the functions of a healthy cell. Defects or loss of function in either of the proteins will result in excessive buildup of defective mitochondria in the cell, leading to diseases such as Parkinson's disease.
Recently, a pathway independent of the PINK1 Parkin pathway was discovered. It was found that the loss of iron also leads to the eventual removal of a damaged mitochondrion. How does iron loss lead to mitophagy? How is this finding so significant? Iron is an essential component that is required by cells for a series of metabolic processes. The mitochondria produces iron-sulfur clusters and heme groups required for many mitochondrial and cytosolic enzymes. A deficiency in iron impedes the ability of mitochondria to form these clusters and heme groups that have an affinity for oxygen. This results in hypoxia, a condition where oxygen levels are low.
In response to hypoxia, a transcription factor HIF1-alpha is switched on. HIF1-alpha induces transcription of genes involved in processes that help to promote delivery of oxygen to regions where oxygen levels are low. Persistent hypoxic conditions eventually result in the destruction of the mitochondria due to its dysfunction.
We predict that impaired mitophagy is going to be a contributor to disease. It's been implicated in, obviously, Parkinson's disease, but also other forms of neurodegeneration, cancer, liver disease, all sorts of things. These turnover mitochondria, have been implicated. So, in theory, it's a good target therapeutically. Can we hit mitophagy, either inhibit it or enhance it? So, this is kind of a long-term goal. This is what we would like to be able to do, to pharmacologically modulate mitophagy, but to be able to do it specifically, not affect all these other processes that are going on. But to do that, we really need to know or identify the specific molecular components that are involved in this process. So then we can potentially design drugs to target these to modulate mitophagy.